So they get a little bit longer time. They get 25 minutes instead of the usual 20. So I think I'll get out of it. There's also where they have more names. So this is Cole Hunt, Sebastian Fisher, Thomas Keeling, Cal Nielsen, Adam Ratana, and Ryan Smithers. Take it away.
I'm Cole Hunt, this is Thomas Keeling and Cal Nielsen, and we are the Peptide Patrol.
I'm Sebastian Fisher.
I'm Ryan Smithers.
I'm Adam Ratana. And that's what we are.
So we are protecting therapeutic peptides from digesting. So let's start with 3 basic questions that guided our presentation, or our project. First, what is a peptide? Second, what is a protease? And third, and most importantly, can we slow down proteolysis? So let's start off, what is a peptide? Well, peptides are like proteins, which you've heard about earlier in the night, but they're smaller, basically. And insulin is a very important peptide. It's essential to the treatment of diabetes. And as you can see here, it's a long chain made up of many different parts, and those are all amino acids, each with a unique identity as shown by the different letters. So what is a protease? A protease is an enzyme that digests peptides, and the one we'll be talking about tonight is trypsin. So trypsin likes to cut at the amino acid lysine. As you can see, it breaks down insulin into several useless parts, and therefore halts insulin from being digested through the digestive tract.
So the question is, can proteolysis be slowed down? Sorry, Mike. So currently, insulin is actually injected directly into the bloodstream. This is because insulin is actually broken down by protease found in the digestive tract. So, in order to solve this problem, we are inspired by prior research, which is showing that if we increase the density of peptides, or increase the amount of peptides, it is able to actually protect itself from the trypsin. This is because if you increase the amount of peptides surrounding each other, it becomes such kind of like a web and entangles each other, which causes a lower chance for the actual protease to find the critical amino acid for proteolysis to occur. And so, current problem with actually analysis with proteolysis is that naturally it has no different features from the before and after the actual reaction. As seen here, they are both blank and look exactly the same.
So the question is, how do we see proteolysis if they're clear at first hand? Now, the answer to this is something called fluorescence, and let me explain what fluorescence is. If a solution is fluorescent, then under UV light, it produces light. No, go back. The peptides we used were not fluorescent before proteolysis, as seen here, but after proteolysis were fluorescent. And what this means is it gives us a way to see if proteolysis even occurred. Now, how does this work? Now we first start with the peptides before proteolysis occurs. And as you can see here, there's no fluorescence, there's no light being emitted. But if we add our enzyme and proteolysis starts to occur, the enzyme starts to cut the peptides and light becomes emitted. Now, why does this happen is the question. Well, we use 2 peptides called the red peptide and the green peptide. Now, the green peptide can be seen as a sun, and why is this? Alone, the green peptide, it fluoresces, it produces light just like a sun. However, when green and red are bound together, then no light is emitted. And why is this? Because the red blocks the green from emitting light, just like a cloud would do to a sun. Now, for all the scientists out there, these are the molecules. EDANS is what makes green fluoresce, and dapsolysine is what makes red block EDANS from shining.
So our actual peptides we use, we gave them these names: red 5E7, red 6E7, and red 7E7. So you can see here there's a backbone on each of them, and those are the red peptides, the ones that contain the cloud that stop the fluorescence. And as you can see, with each one, there is more sun E peptides attached to each backbone, increasing the peptide density. So what our E peptide actually is, it's our EDANS glutamate, which contains that compound that fluoresces under UV light and is the actual sun. Lysine is an amino acid that trypsin likes to cut at, and acetylysine is the amino acid that allows us to connect it to the backbone. And this is what our E peptide looks like in our solution. And this is what it looks like under UV light, and you can see it's like nice and glowing. So these are 3 different red peptides we use: red 5, red 6, and red 7. And as you can see, each one includes a greater number of propargylglycines, which is the points at which it can connect to an E peptide. And the dapsolysine is the cloud amino acid that quenches the fluorescence from the EDANS. So this is what it looked like when we were synthesizing our red peptides on a column. This is what it looks like after we dried that peptide and it was nice and fluffy down in this vial. And this is what it is when we suspended it in an organic solvent. So how do we actually attach these peptides? How do we go from this to that?
That's called click chemistry. So click chemistry begins with what we call an azide and an alkyne. Azides are characterized by 3 nitrogens all connected, and alkalines are characterized by a triple bond between carbon atoms. In order to make a click reaction happen, you have to add a copper catalyst, which you can see right here. When a click reaction occurs, it forms a trizol, which is the connection of the azide with the alkyne. So, in our case, we used the red 5 peptide, which has propargylglycine, our alkyne, and the E7 peptide, which has acetylysine, which is the azide. And when we add copper, it allows this reaction to occur, forming our click product. So the question is, can our group click peptides together? So here we have a video showing the actual process for clicking peptides together, if it works. I think it will. So we have our red 5, our alkyne. We're adding it to the reaction vessel. Now here's our E7, adding it to the reaction vessel as well. We add something called sodium ascorbate to the reaction vessel to help the reaction run. But before adding the copper catalyst, we collect a T=0 sample before the reaction actually starts. Then we add our copper sulfate, which is our copper catalyst, to the reaction vessel. We mix, and the reaction should occur.
So, how do we know that this works? So we use our procedure during our click where we in our single click reaction, we equal the red and the 7 peptides together. And here is an HPLC, which Dickerson's group went over, where it basically separates molecules out over a time map. And as we can see here, we have our red and our E. And then after we do our run experiment, which in this case was 4 hours, we can see a distinct peak in between those two and some red left over. And here is
Well, how do we know that this middle peak was in fact our clicked peptide? Is we did mass spec, which is also something that Dickerson's group went over. And we can see here that this is our mass of our clicked molecule, which we calculated to be about 723 molecular weight units, and it was that when we took a reading of it. So in our red 6 E7, since it is double clicked, we increased the amount of E7, making it a 2-to-1 ratio, E to red. And again, we can see our two distinct peaks, our red and our E, and then finally our center peak of our two clicked. And then again with our red 7, E7, this time it is a 3-to-1 E to red ratio, ending up in, again, the two distinct peaks before and the one distinct peak after. This time we had no red and instead had an excess of E.
Okay, so now the big question is, do our click peptides slow digestion? Did 3 years of experiments come to fruition? Did we accomplish what we set out to do? Alright, so to find that out, we have to run, we talked about proteolysis earlier, the digestion of peptides. We have to run proteolysis, we have to run it, and we have to measure the fluorescence as the proteolysis runs. So we do this, as you're about to see, to get it to work.
For chemists, it's not computer scientists.
So we do this on a machine called the fluorescence spectrometer, which basically measures the fluorescence of a solution. And our solution we create by first adding phosphate buffer right here, which is just keeps the pH at a neutral level so it's not acidic, and that allows proteolysis to run. And then we add, this is our click product here, so we do this for each one, so we'll have single click, and then we'll do one with double click, and then the triple click. We mix this around, and then we'll put it in the machine. Then while it's in the machine, we'll add trypsin here, which we mentioned earlier is the enzyme which starts proteolysis and digests the click. And then on the machine, over time, we'll start to see here readings of the fluorescence, and as expected, it will increase because as the trypsin digests the click peptides, that sun peptide will begin to fluoresce as it's separated from the red, which is the cloud. Okay, so these are the results that we got from the single click, the double click, and the triple click run using that proteolysis method. So as you can see, they all increase to about the same fluorescence, but the important thing is here what the rates were at which they increased in fluorescence, because our hope was that the triple click would have a slower rate than the other, or that the single click would have the fastest rate, the double click would then be the next fastest, and the triple click would finally have the slowest rate of proteolysis. So when we manipulate the data to find the rate at which concentration decreases, we did in fact find that we were successful, and the single click has the slowest rate, the double click is the next slowest, and then finally, or fastest, next fastest, and then finally the triple click is the slowest rate. So we were successful in slowing down proteolysis. So just a quick recap, we successfully synthesized Red 5 and E7 peptides. Then we clicked E7, the green peptide, to Red 5, Red 6, and Red 7. And after that, we measured the proteolysis rates and we displayed that the proteolysis rate decreases as the click density increases. So now,
So with this conclusion, we successfully repeated essentially the data found in previous experiments that we showed earlier, Gianesky's. However, we made it much simpler by using clicked peptides. And this is basically a high school level chemistry concept that allows the cheaper and quicker production of these higher density peptides, which protect each other from digestion. And so for next year's groups, they might try different, more novel shapes of peptides in order to increase the chance of the trypsin not being able to find the actual lysine, making the peptides more complicated and tangled together. But for us, that's it, so thank you.